Hello. I hope everybody's doing well. I'm just setting up here. Um, as usual, if anybody wants to drop in and ask questions, we'll deal with those. And I just wanted to say a few words about the exam next week. Your next exam is coming up on Tuesday. So that's what we'll do today. So you have 3 topics on the exam next week. The first one is prokaryotic cells. The second one is eukaryotic cells. And the last one is acellular microbes, which includes viruses and prions. Of those 3 lectures, it's the third one that students find most challenging on this exam. So make sure you give that lecture about acellular microbes all the attention that it deserves. It's a new subject for most of you, viruses, and some of the terminology can be a little bit confusing. It's important that you know the vocabulary that we use to describe viruses. It's important that you know terms like capsid and capsomere and nucleocapsid, spike protein. It's important to know the 2 types of viruses, the naked viruses and the enveloped viruses, and which types of organisms get which kind. It's important to know that all living things on our planet are susceptible to some kind of virus. Even little bacterial cells get infected with viruses. And of course, the kind of virus that they get infected with is called a bacteriophage. We talk about the shapes of viruses in that lecture. Remember, some viruses are helical or sort of rod shaped. Some of them are polyhedral. They're made out of a whole bunch of flat plates that are all assembled together. Some are spherical. Our friend SARS-CoV-2 is a spherical virus, so is the influenza virus. And some viruses have a very complex shape. The bacteriophages are a great example of a virus that has a complex shape. They're just more complicated in their shape than the other viruses. The giant viruses also have a very complex shape. We talked about the fact that viruses are named in a very sort of scattered way. We went down a list of the ways that viruses are named by things like the disease they cause, or the geographical location where they were discovered, or by the name of the scientist who discovered them. Sort of a disorganized system for naming viruses. But we do have a system for classifying viruses. If you remember, that's called the Baltimore System. And the Baltimore System classifies viruses according to 4 features that the virus has. I'm not going to ask you a lot about the Baltimore System. I just want you to understand those 4 things, the 4 things that are classified for a virus in the Baltimore System. I'm going to pull up a screen here. If you'll just bear with me for a second. So what you should be seeing on your screen right now is the title slide for the acellular microbes lecture. And I'm going to just go down through the slides till I get to Slide number 22 about the Baltimore system. This is the slide that has the list of things that we need to know about the Baltimore system. There are 4 things that a virus gets classified by. Number 1, the kind of genetic material it has, whether it's DNA or RNA, and whether it's single-stranded or double-stranded. Number 2, the sense of any single-stranded genetic material. And remember, sense is really just a designation that is used to describe the direction of twist in that molecule. That's all you really need to know about sense. We describe single-stranded DNA or RNA as being either positive or negative sense. The third item on the list is whether or not the virus uses an enzyme called reverse transcriptase. And the last one is how the virus is replicated by the host cell. What is the mode of replication? What is the process, in other words, that the host cell uses in order to copy the viral genetic material? So those are the 4 things that you have to know that the Baltimore system will classify. If we back up a couple of slides, remember we talked about how influenza viruses are named. Remember, there are 4 types of influenza viruses. It's A and B that we really think about clinically in human medicine. A type is the really important one in the sense that it's influenza A viruses that can cause pandemics. Influenza B viruses also cause disease, but they're not as problematic as influenza A. It's the influenza A viruses that are classified according to that H and N numbering system, as well as a list of other features of the virus. So remember, it was the WHO, the World Health Organization, that came up with an international system for classifying influenza viruses. We start off with the type that it is, A, B, or C. Remember, the D type only infects cattle, so it does not infect humans. The host of origin, in other words, what animal that particular influenza virus came from before it started infecting humans. The geographic location, the strain number, the year of isolation. And again, for influenza A, the H and N numbers that belong to that virus. So most viruses are named in kind of a haphazard way. But influenza does have an internationally accepted system of naming. And we use that system so that we can talk about influenza in public health. We can talk to our friends who are in public health in other countries, and we all know which virus we're talking about. We have to work together in public health with other countries around the world, particularly when it comes to viruses like influenza, which can be so very problematic in terms of the number of infections every year and the number of every year. The SARS-CoV-2 virus, remember, it's not an influenza virus, it's a coronavirus. And so it falls into that sort of mishmash of naming. Part of its name describes the disease it causes. And part of its name describes the way it looks. SARS stands for something. It stands for severe acute respiratory syndrome. That's the disease, right? Or at least the first part of the disease that this virus causes. The CoV stands for coronavirus. And of course, coronaviruses got their name, remember, because when someone looked at a coronavirus under a microscope, the spike proteins coming off of it reminded them of a crown. That's all corona means. It means crown. And the reason it's SARS-CoV-2 is because there was an original SARS virus that came about in the early 2000s, did not cause a pandemic, but certainly caused a bad epidemic in parts of Asia and in Canada. So most viruses are named according to some feature of the virus or where it was discovered or who discovered it, kind of in a haphazard way. Influenza is named more formally, has a more formal way of naming it. So as you're preparing for the exam and you're going through the material for prokaryotes and eukaryotes and acellular microbes, know that the exam will be split approximately equally between those three topics. So about a third of the questions will come from the prokaryote material, about a third from the eukaryote material, and about a third from the acellular microbe lecture. It'll be about the same length as the last exam was, and about the same level of difficulty. Remember, you can take it any time between 8 o'clock in the morning and 8 o'clock at night. You just need to get it to me by 8 o'clock at night, unless you've made an arrangement with me to submit it at a different time. Take your time. Take your time. Read the questions. Make sure you know what's being asked before you answer. And of course, read all the possible choices before you choose the best one to answer the question. Okay? Don't make the mistake of sort of quickly reading the question and then reading the first answer and thinking, oh yeah, that's the right answer, and checking it off before you even read the rest of the answers. That's not a good strategy when you're taking a multiple choice exam. There will be at least one bonus question, probably two on this exam. Please try the bonus questions if you have time. You can't hurt your score. Bonus questions are, if you get them wrong, it doesn't hurt you. And if you get them right, you'll get a couple extra points. All right, that's all I wanted to talk about today regarding the exam next week. If any questions come up while you're preparing the material, please feel free to message me, and we can talk back and forth over email about them. I got a couple of emails earlier about a question on the quiz that you had this week. I was glad to see that. Please ask questions if something doesn't make sense. I actually had a couple of students ask about one particular thing, and I thought I would mention it today, just in case you're not aware. The terms cell membrane and plasma membrane mean exactly the same thing. It's a little hard because sometimes you'll read a certain textbook or you'll read a particular website and they will refer to the membrane that surrounds the cell, so the outer membrane. They'll refer to it as the cell membrane. But when you go into other sources, other books or websites, they'll call it the plasma membrane. So it can be a little confusing, but those two terms mean exactly the same thing. They refer to the membrane that surrounds the cell. Remember, in eukaryotic organisms, there's membrane inside the cell as well. The membrane surrounds the organelles. Some organelles have 2 layers of membrane and some organelles have 1 layer of membrane. There's lots of membrane inside a eukaryotic cell. All cells have membrane surrounding the outside of the cell. That was a good question. I'm glad a couple of you sent me messages to ask about that. Good questions. All right, everybody, that's all I wanted to talk about with you today. Again, if you need anything, please message me. Don't hesitate to message me. Good luck with your studying. I hope everybody's doing well. I hope everybody is feeling well. And I will talk to you again soon.